Hey Team Bio, welcome to your screencast on denaturation of proteins or enzymes and enzyme inhibition. Okay, so you know and love the induced fit model of enzyme action. Here we have hydrolysis. We've got our enzyme sucrase, which is going to bind the substrate sucrose. The binding is going to actually change the shape of the enzyme. Remember, we have this conformational change when the enzyme binds the substrate. Here we have H2O coming in. The enzyme puts the substrate in the exact right position to catalyze the reaction, lower the energy of activation, and then we get our products, glucose and fructose, and the enzyme returns to its original shape, so it's ready to do the reaction again if it has more substrate, sucrose. Okay, so you know and love the induced fit model. Today in class, we saw some examples of when this stopped working. We had a functional folded protein. In class, we were looking at the function of the enzyme catalase. And then we did something to impact the function. We denatured the protein. We went from a complex three-dimensional folded structure to a not very complex unfolded. I wouldn't say it's an unfolded protein. Let's change that. Let's say instead, more accurately, I would argue, we changed it back to a polypeptide. Okay, how do we do that? Well, you know that this complex structure of here is a beta sheet, and this is an alpha helix. We know that this is a tertiary structure protein because it's composed of one subunit, but it's got these multiple interactions holding it in place. This is a combination of hydrogen bonds, hydrophobic interactions, and disulfide bonds holding this structure in place. But the primary mechanism that this shape is held in place is through hydrogen bonding. So we have, for example, hydrogen bonding holding these in place, these structures, the alpha helices and the beta sheets in place. But there's also going to be hydrogen bonding holding this in place, and holding this in place, and this in place, and this in place. And also, yes, hydrophobic interactions between the R groups and disulfide bonds. But the major contributor to holding protein shape together is going to be these H bonds. So how did we denature the protein, turn it back into a long, straight, linear polypeptide? Well, we can do it in a bunch of different ways, but they all have to do with disrupting the hydrogen bonding that creates the structure of the protein, and anything that breaks that hydrogen bonding is going to denature the protein. So one of the first things we saw in class was that we changed the pH. We added a strong acid. When we added a strong acid, we actually increased the H+ concentration in solution. Now, most proteins are functioning and take on their folded pattern in a neutral environment. And when we add a lot of free H+ in solution, now this hydrogen ion can come in here and it can bust up this interaction. So now we have an H+ ion here forming a hydrogen bond here instead of the hydrogen bond forming across here. And so this little section of the protein falls apart. And then the same thing happens over here. This bond is broken, and now we have a new hydrogen bond to an H+ ion in solution. The same thing is true if we add a strong base. A strong base decreases the amount of H+ in solution, but it increases the amount of OH- in solution. So similarly, if we had a hydrogen bond across here, the OH minus is going to come in, break that bond up, and instead form an interaction with this side of the protein, still breaking this bond. And likewise, if we had an interaction across here, the OH minus could come in and form a bond with this side, breaking up that interaction. So add enough OH minus or H plus and the protein will fall apart. The same number of hydrogen bonds exist, except now those bonds are not intramolecular. Now they are intermolecular. They're bonding with ions in the solution rather than within the molecule itself, or the compound, the organic molecule. Okay, so changing the pH will denature the protein. Another way to denature a protein is to add lots of ions. So, for example, if you added lots of salt, which would be Na plus and Cl minus, that would do the exact same thing as H plus and OH minus. We didn't do that in class, but we could have added a bunch of salt to solution, and that would have denatured the protein as well. Another thing that we did in class that denatured the protein was that we changed the temperature. So, if a protein gets too hot, this will break the H bonds. So when we heated up the catalase, when I boiled the liver, what happened was all of these H bonds that are holding the structure together, they broke. But the covalent bonds that hold together the primary structure, they did not break because we didn't burn the protein, we just boiled it. It didn't get hot enough to break the linear structure, the peptide bonds that hold this polypeptide together. It only got hot enough to break the hydrogen bonds, which are slightly weaker interactions. But we still denatured the protein when I boiled it. The last thing we did was we froze the liver. And this froze the H bonds into place. But that actually, it didn't stop the reaction entirely. What you saw was that this shape shifting that the enzyme had to do, this conformational change that it had to undergo, was slow at first because the bonds that were holding the structure together were really rigid. They were frozen in place. So we saw like really slight fizzy. So the reaction was proceeding really slowly at first. And then over time, you saw the bubbles get bigger and bigger. The reaction sped up over time because it's an exergonic reaction, the reaction that catalyzes. So it released heat. So we started out with an enzyme that was frozen, and it was really slow to undergo this conformational change. But then as it catalyzed this reaction again and again, every time it did that, it released heat and it warmed up the enzyme. So we saw the enzyme cycle faster and faster as it defrosted itself. But yes, you can freeze an enzyme, which isn't going to destroy the enzyme. It's just going to freeze these bonds into place and make the reaction that it catalyzes much slower. Okay, so I guess this is a star. This doesn't denature the protein. It just freezes the bonds into place. Okay, so protein denaturation is generally irreversible. Whoops. The protein shape has been destroyed and it is no longer functional. However, there are some ways to inhibit the action of an enzyme that is a reversible interaction. Okay, so here we have an enzyme with a substrate, and you'll notice that the substrate fits perfectly into the active site of this enzyme. We've been talking about this a lot, but about how the enzyme active site is perfectly fitted for the substrate, but you should know that some people call this a lock and key kind of fit. Okay, so there are two basic types of inhibition. The first type of inhibition is called competitive inhibition, and this is where the inhibitor binds to the active site. And prevents the substrate from binding. So it physically blocks the substrate and doesn't allow it to bind to the active site. A non-competitive inhibitor, also sometimes called an allosteric inhibitor, binds to a site far away from the active site. But in binding, it changes the shape of the active site such that the substrate no longer fits. So this is a non-competitive or allosteric inhibitor. Your body actually uses these two types of inhibition all the time to regulate enzymatic processes. And let me give you an example of this. Okay, imagine we have a enzymatic pathway. We have our starting molecule A, and eventually we want to turn it into our product D. But in order to get to D, we need to convert A to B with enzyme 1, and then B to C with enzyme 2, and then C to D with enzyme 3. So we're going along converting A to D in that way. But potentially, we don't want too much of this product D. So what happens is that D is actually an inhibitor of enzyme A. So when the amount of product D gets to such a level that it now comes in and binds to enzyme 1 and prevents A from converting to B. And then so forth. If we don't have a lot of B, we're not going to make a lot of C, therefore we're not going to make a lot of D. And so our levels of D will fall. When the levels of D fall, now there's not a lot of D product around to inhibit this enzyme 1. And so enzyme 1 now can start converting A to B again. So the levels of product D will increase once again until there's so much of it that it inhibits enzyme number 1 again, and then the levels of it fall. So this is a way to mediate levels of protein products or just products in our bodies through this what's called feedback inhibition. And this would be an example of a negative feedback loop. And this helps maintain homeostasis. It keeps levels of things buffered within certain limits. So that's one mechanism of how enzyme inhibition is used by your body. Additionally, many drugs and poisons work through enzyme inhibition. So, for example, caffeine. Caffeine is an inhibitor that blocks adenosine receptors in your brain. And adenosine is a molecule that kind of indicates fatigue. So when caffeine prevents adenosine from binding, those fatigue signals that your brain should be receiving, because adenosine is building up, are no longer recognized by your body, which is why caffeine gives you a feeling of increased energy and alertness. It's blocking the adenosine receptors. Ibuprofen. Ibuprofen blocks prostaglandins, and these are chemicals that promote pain and inflammation and fever. So if you block the production of prostaglandins, you will not have the inflammatory response of pain, more liquid secretions aggregating in a certain place, swelling, and fever. So ibuprofen is another drug that works on enzymes as well. Chemo drugs for cancer often target enzymes that are responsible for the cell cycle and cell reproduction. So cancer is uncontrolled growth of a certain type of cells in your body, and so chemo drugs target reproduction, cellular reproduction. Antibiotics. Target enzymes in bacteria. Sometimes they target ribosomes of bacteria. Sorry, brain fart. And sometimes they target the enzymes that are responsible for building cell walls. All sorts of different antibiotics target different bacterial enzymes. An example of a poison would be carbon monoxide. Carbon monoxide is carbon bound to oxygen, and it binds to hemoglobin in your red blood cells and prevents your red blood cells from transporting the oxygen that you need. These are examples of a bunch of different drugs and poisons that act as enzyme inhibitors. Okay, that is all. See you in class tomorrow.